Hello and welcome to the Microbiome Medics podcast. I'm Dr. Siobhan McCormack.
And I'm Dr. Sheena Fraser, and we're your co-hosts.
We are both GPs and lifestyle medics with a shared passion for microbiome science. We'll be translating the evidence and packaging it into actionable, bite-sized chunks so that you can harness the power of the microbiome to improve your own health and that of your patients.
Hello and welcome to another Microbiome Medics podcast. I'm Dr. Sheena Fraser, and today I'm joined with Dr. Michael Cox. Dr. Michael Cox is going to introduce us to the lung microbiome today. So we've already had a little intro into the vaginal microbiome, the skin microbiome. We've obviously touched a lot on the gut microbiome. So today I'm really excited to delve into the lung microbiome. Dr. Cox is based in Birmingham. And his research focuses on microbial ecology of the respiratory tract. And he's translating the microbial analysis of the respiratory tract into clinical, useful, knowledgeable information for our clinicians. So today, let's have a little chat. We're going to touch on what the lung microbiome is. We're going to take you through the importance and the correlation between the lung microbiome and the gut microbiome, and also the communication between the two, and then translation into the clinical side, how this impacts on our health, how it impacts on disease, and go through from there. So thanks, Mike. Thanks for joining me today.
Thanks
Welcome to the podcast.
Thanks.
So, tell me a little bit more. So I'd like to really start with is, first of all, how did you get into studying the lung microbiome? Because there's not that many of you around.
It's not. We're a weird bunch. So my route was essentially a sort of field change, really. So I started out studying seawater microbial ecology. So communities of bacteria in seawater and their impact on climate, and particularly the ozone layer at the time. And how they use trace gases. So interactions between bacteria and their environment. And then around 2008, I took up a position at the University of California, San Francisco. And the techniques we were using in the environmental field were suddenly starting to gain a bit of traction in the human microbiome field or in human research. And I got the opportunity to study people with cystic fibrosis, or at least the samples from people with cystic fibrosis, which has always been known as a sort of polymicrobial infection in the lungs, this chronic lung disease. And so it was really a techniques-based thing. So rather than a bucket of seawater that I was studying and the interactions with its environment, I was studying a bucket of sputum instead.
Lovely.
This is the field. It's a lot of sputum is what we end up with in the lung microbiome field. So I came following the techniques really across in that route, and then that's expanded to other diseases and healthy lungs over the course of my research.
Lovely. Okay, so let's think about the lung microbiome. And I'm really interested in how the lung microbiome compares to the gut microbiome and how it differs. So Can you give us a little bit of an info on that?
So I think, it's quite different. The key thing is that as a site, we're constantly clearing our lungs. So we've got the mucociliary escalator, we've got sort of rafts of mucus, and that's designed to keep things coming out. But we do also have a constant flow of stuff going in as well. So we've got sort of microaspiration from saliva. We might have bits of the oral microbiome and the nasal microbiome traveling down into the lungs. And that's normal. So probably there's quite a bit of microaspiration that goes on overnight. So it's more of a balance between these two processes of clearance and deposition, at least in healthy people. So the numbers of bacteria that are there are much, much lower than you see in the gut. That doesn't necessarily mean they're not doing anything, which I think is super important because, quite low numbers of bacteria can have quite an impact biologically because they do all sorts of unique things that human cells don't do. So it's this sort of balance, this equilibrium between what goes down and what comes up. We have only the one hole in the lungs as opposed to two. It's not the donut of the gut. So clearance is a slightly different process. But yeah, numbers are different. Composition is a little bit different as well. So the most common organisms we find there are quite different to the most common organisms that you'd measure in the gut microbiome. So the sort of top favorites are things like Streptococcus, Prevotella and Veillonella, which you can find in the gut, but not at the same abundances. So those top three are the most abundant organisms in everyone.
And are those organisms, they're aerobes, so organisms that like a bit of air, or are they anaerobes?
Yeah, they're weird. So what we find actually is usually you would consider microbiologically Prevotella and Veillonella in particular to be strict anaerobes. They're quite hard to isolate aerobically. You'd have to usually use an anaerobic cabinet to grow them. But we find they're the most common organisms alongside Streptococcus. And I think that's sort of explainable by, the lung surface is probably enriched in CO2, about 5% CO2. If you measure right at the lung surface, you've probably got less oxygen than, in ambient air. So maybe these organisms, we could consider them microaerophilic. That's a sort of microbiology term for this kind of state, where they perhaps can tolerate a little more oxygen than their sort of strict anaerobic expectations of these bugs. But yeah, it's sort of counterintuitive that an organism designed for use of oxygen at the surface has these anaerobic organisms.
Interesting. Okay. And so they're similar in that there's a flux of organisms.
Yes.
But obviously in the lungs, that flux goes down and then back up.
Mm-hmm.
And then in the gut, obviously it goes out and then is expelled out the other end. Yeah. Okay. And then with the microorganisms in the lung, obviously you've got fewer of them. Is it variable in different parts of the lungs? So, how do the end pieces, the alveoli, the little air sacs right at the very end, how do they compare to the big tubes, the trachea, the bronchi? How do they compare?
This is really difficult because one of the challenges we have in the lungs is sampling.
Yeah.
So we're always sampling, usually through the mouth or nose. So anything we find is not independent of those two sites, usually. There have been some studies of explanted lungs. So in people who've had to have lung transplants, then some groups have looked at the lungs that were taken out. And that allows you to sample through the side and to be very careful that you're not contaminating any part of the lung with any other sort of microbiological source. And there they can see differences around the lung. So different parts of the lung might have different bacteria, different compositions of the microbiome present. The caveat there is that's always end-stage lung disease because you don't get a pair of lungs that are good lungs that you don't want to use for a transplant or some other purpose. So it's an end-stage disease. So we don't really know, I don't think, in healthy people about variation like that. What we can probably say for sure, I think, is the further down the tree you go in the lungs, the fewer organisms you would expect to see. I think they do reach the alveoli. So, we have another clearance mechanism there, the alveolar macrophages that sort of roam around gobbling up bacteria and particles. And they wouldn't be there if they didn't have a job to do, and these sort of sizes of particles reach down there. But probably there's a gradient of higher numbers at the top in the larger airways, and then fewer in the peripheral airways, and then fewer still in the alveoli. Okay.
And you mentioned this flux from the oral microbiome and the nasal microbiome, so all the little microorganisms in the mouth and the nose, and how they can travel down into the lungs. So we're sort of microaspirating, so just inhaling little pieces of these microbiomes into our lungs all the time, and they're traveling down. So So how does that relationship between the nose and the mouth go? And what is the importance of that, would you say?
This is sort of interesting. And I think inhaling as well, because the sources of the organisms, so Streptococcus violinella, Prevotella, we certainly find in the oral cavity as well. Those probably not the major organisms in the nasopharynx, in the nasal cavity. So there's certain things that are unique to these environments as well. So the nose, you often see things like Dolosigranulum and Corynebacterium. I love some of these names. But we don't see those routinely in samples from the lung or lower down in the lung. Similarly, there are a lot of organisms that are associated with dentition, with teeth, with sort of crevicular fluids, that fluid that builds up between your teeth and your gums, that are quite unique to that site that we don't see in the lungs as well. So there's some process of selection going on here. There are things that are either easier to get into the lungs or tend to stick around a bit there. And that's a very hard thing to measure. Partly because of how you sample, and sampling over time is what you need to do. But probably there is something like that, because across different people, we always see those three dominant organisms in health. So there's something about that environment that allows them to be there. We don't know what that is.
Okay, so they're the travelers.
Yes, yeah, I think so. Resident would be a controversial term, because you've got this process of them moving out. We don't even know whether there's growth in there. So in the gut, for example, it's repopulated because, there's bacteria definitely growing in your gut. We don't know whether the ones in your lungs are actively growing in situ, which would shift that balance as well.
So then maybe we should talk about what they're actually doing there.
Hey, that's my job. That's the $1 million question. Yeah.
So what are these bugs doing in our lungs? What is the purpose?
I just think they're sort of. I always like to think of humans as sort of meat puppets, and I'm all about the bacteria when it comes to this. But they're just hanging out, having a good time. Streps. Probably the Streptococcus is, existing on the mucin that we produce. And that's produced constantly, and as well as being removed, probably being degraded by the Streptococci as well. I think probably Prevotella and Violinella have roles there as well. So they're sort of happy grazers. Mm-hmm. Positive impacts are sort of hard to show. So there's a little bit of evidence that certain organisms might interact with inflammation. So there's one called Rothia mucilaginosa that some people have shown in vitro, at least in lab experiments. Some strains of it can impact inflammation. But this kind of research in the lungs, it's more common to talk about those kind of interactions in the gut microbiome, because it's a tough site to sample and to understand. That research lags in the lungs. So there might be interactions there that sort of dampen down inflammatory responses, but there's certainly plenty that increase inflammatory responses, because we know about the sort of lung infection pathogens and stuff.
Yeah, and so we know from the oral microbiome as well that they can play a role in your initial fighting off of infections. So some of the more mucus dwelling bacteria, they play a role with IgA, like lactobacillus plays a role with IgA. Are there any lactobacilli in the lungs?
Yeah, you can detect them. We don't know what they're doing. The abundance isn't very high, because it's a low abundance environment altogether, and then they're not the most common ones, but you can detect them. But similarly in the gut, they're not necessarily the most abundant ones. I think probably of these kind of sort of, we call them commensals, where they're there, but we don't know what they're doing, and they might not be having a positive or negative effect. They might just be sort of hanging out. I wonder whether the streps are going to be those abundant ones are going to be more important in terms of interactions with us, because they do produce, Streptococcus produces lactic acid, lactate, as a normal part of its metabolism. It is technically in the larger group of lactic acid producers, of which lactobacillus is a relative. So, you can detect it. We don't know, again, what it's doing. That's going to be a common theme, I think, when I'm talking about the microbiome.
I know, absolutely. I did come across a paper that looked at the impact of some probiotics on lungs, and Lactobacillus helvetius helvetius, which is not one I'd heard of before.
No.
Evidently has a positive effect on the lung microbiome by increasing IgA in the mucosa, which is sort of an early defense against respiratory pathogens, which is interesting. But I don't know whether I don't know where you get helveticus from.
No, and it's really difficult because lots of the ways we measure these things are not very good. So a lot of the sequencing techniques that we have to use, so you can isolate bacteria, but we don't isolate all of them because you have to use a huge range of conditions to actually isolate them. So we're always sort of subsampling when we try and grow something because we will be missing a bunch of stuff. So we use a lot of DNA sequencing techniques. The issue with those is identifying the organism. So firstly, knowing which one it is, because maybe we'll only get a genus name, but we know that the impact to probiotics, we know that pathogenicity is definitely a strain or substrain level property. So we're not accessing that information. We're just we're finding the village. We're not finding the address. So there's that issue. The other one is that the sequencing techniques have this property that they are relative abundance methods. They're not quantitative. So on the whole, what you do is you sequence, and it's a proportion that you see. If something increases in abundance, we don't know whether it's increasing itself and everything else is staying at the same level, or it's increasing and we're pushing everything else out of the ability to sample it. So that makes everything really difficult. So we can usually see if something has increased in abundance. We can't see decreases with this method. So it's where that becomes particularly important for understanding interactions between organisms is that we can't see negative interactions. People will assume them because we think of it in competition. If something's increased and something else has gone, we think, oh, they're in competition with each other. But actually the technique can't tell us that. We need a real quantitative technique. So if you're reading papers about this, what you'd like to see there is somebody do a specific qPCR or a specific sort of isolation of those two organisms to confirm whether they're genuinely in competition with each other, or in fact, it's just that method hasn't been sensitive enough to detect the change or lack of change in one of them. So yeah, it gets that sort of competition thing, which has got to be essential to understanding infection and protection against infection, is something we are not good at generally with this technique. So we have to be quite careful.
And I guess from that technique, we're sort of assuming function.
Yeah.
Whereas, now we should be looking at function a little bit more, rather than just considering who's there and how many of them are there. What are they actually doing and what are they producing? What are there any studies on function of these organisms in the lungs?
So in the lungs, far fewer, because it's hard to look at function in situ. It's really interesting to me because this parallels all the questions we had in environmental microbiology 20 years ago. So we started by trying to profiling what was there, and then associating that with measures in the environment. So in the human sort of study, we're measuring what's there and associating that often with disease.
Yeah.
Disease is really actually a far down the line process. Mm-hmm. It's not the what's actually happening. It's not what the organism is experiencing. It's not what the cells are experiencing. It's the ultimate endpoint. A lot of life has happened alongside that. So those associations, I think, are going to be, at the moment, they're quite distant. A lot of people measure lung function in microbiome studies, trying to find an organism or a group of organisms that relates to lung function at all. Usually it's negative. However, that doesn't mean it's not important. It means we're potentially measuring something so far distant from what it is, the processes that are actually going on. In vitro, so you can look at function of organisms. We're quite good at sort of measuring. You can use models to look at how virulent they are. You can use models to look at whether there's interactions with the immune system in some way. Mm-hmm. Those models are always a compromise. They're always something slightly different to what's going on in the human. So one of the things I think lots of people in this field are trying to do at the moment is to try and find good models to answer particular questions. And that's everything from, just changing the media that you're growing the bacteria in to make it look a bit more like an airway. So literally adding some pig lung mucin to that, or pig stomach mucin to your culture to see how it interacts with mucin, to models that are sort of organoids, where you build up a 3D cell culture where you start to see ciliated cells that you would expect to see in the lungs as well. That's so intensive that, adding bacteria to that is a risky maneuver, because you get it wrong and you've ruined your cell culture as opposed to achieved something in terms of understanding the interaction. But yeah, it's what everybody is trying to do at the moment. I'd love to do more stuff that's closer to the reality of the lungs and disease, and so would everybody else.
Yeah, so I'm getting the impression here that it's not an easy subject that you're studying here. This is not an easy field.
It's sort of why I like microbiome, because it's as a topic, it's because it's important. We know it's important. We know that there are direct interactions with people. We know it's probably going to relate to both health and disease, but it's so confounded. There's so many things where maybe we're measuring the right thing, maybe we're not. Maybe it's related to this aspect of disease, maybe it isn't. It's full of red herrings. And that actually makes it quite interesting to try and pick all that apart. So that's why I like it. But yeah, it's a bit messy.
Okay, well, let's talk about things that we can kind of already say about it. So let's talk about the influencers on the lung microbiome. Yeah. I've got a list of things that I think are influencers on the microbiome. Let's go through them, and then you can tell me whether they are or they aren't.
It'll probably be maybe, a lot of maybes, but go on.
Okay, all right. So first of all, pollution.
I think probably, yeah. So we know that there's... So one of the proxies we have for an influence on a microbial community is whether we see any impact on infection. So if there's more pollution about, you've got higher chances of getting infection. We can probably anticipate, therefore, that there's an impact directly on the microorganisms in the lungs. People have shown that pollution and sort of smoking can change how bacteria interact with lung cells. So it might make them a bit stickier, might make them a bit more pathogenic, that changes their expression. So yes, I would say pollution does have an impact there on the microbiome. And quantifying it across all these organisms, because there's quite a few of them at any one time in a population of people, you're going to see a lot of difference there in responses. So there's a lot of other things that interact with that. But yeah, for sure, pollution, I think, is a good one.
And I guess there's lots of stuff in pollution that behaves
It's
differently.
not one thing.
It's not one thing,
Yeah.
exactly, because there's gases and there's heavy metals, and there's particles, soot
Diesel
particles, and there's lots of different things there that will have slightly different impacts as well on the lungs and the microorganisms.
Yeah.
But I would have thought that most of them are pro-inflammatory, and creating some degree of inflammation in the lungs, either through a direct mechanism on the tissues or an indirect mechanism via the microbes or the or other things that it's shifting in the community and in the environment.
Yeah.
Disabling the cilia, the little hairs, doing things like that stops the mucus from traveling up and down.
Yeah. So we know that has an impact using sort of, again, a sort of proxy in disease where cystic fibrosis, for example, mucus is thicker, so the ciliary beat is less effective, and that leads to a build-up of mucus, which gets infected. And there's a sort of related but quite different mechanism in another disease called primary ciliary dyskinesia, where there the mucus is normal, but the cilia don't beat properly, so they're not shifting. Mm-hmm. And there we see similar chronic infections, but actually slightly different combination of organisms. Mm. So I think that's kind of interesting how, ecologically, the environment is a little bit different. So those two things aren't quite the same. And therefore we see slightly different organisms. Yeah. But getting back to pollution as well, I think the other thing to remember is that bacteria are biological entities as well. So if there's something that's a bit toxic that's about, often those things are sort of quite generally toxic or dangerous to living cells. They will affect the organisms too. So we did a population study a number of years ago. So this was when I was based at Imperial College, in collaboration with big population study in the town of Busselton in Western Australia. And there we looked at throat swabs, the back of the throat, which is sort of a good overlap with what's in the lungs in healthy people. So rather than doing hundreds of bronchoscopies across this town of Busselton, we took throat swabs from an ongoing health study. And there we saw a really clear relationship with smoking. So people who'd smoked longer or a larger amount, or had ever smoked and then quit, had different combinations of organisms in their lungs. But the impact of smoking seemed to be that total numbers of bacteria were down. So, there's reactive oxygen species caused by cigarette smoking. There's toxic compounds in cigarette smoke. So they've had an impact on the bacteria, as well as causing inflammation. You've got fewer bacteria present in the lungs. But interestingly, some of them survived a little better. So everything was down, but the streptococci were down less. So in terms of the composition, you see the skew to everything becoming streptococcus. Now, if streptococcus has a particular function, and now it finds itself freed from all these other organisms and in a more inflamed environment, what happens to its function there? Is it still functioning as it was when it was a commensal? Is it now doing something else which may be cycling and helping to increase the inflammation that you have there anyway? Interestingly, we think we saw this. This was, we didn't quite have the numbers to be certain about this, but what we thought we saw as well was that as people stopped smoking, the lung microbiome returned to normal. So it started looking like a healthy one. And the longer ago you quit, the closer to what we usually see in healthy non-smokers was the case. So it's the same as, stopping smoking has an impact, a positive impact on your lung microbiome too.
Did you see a point where it normalized, or was that different for different people? Or
So this is where it gets, because we didn't quite have the sort of range in numbers of age or time since people had stopped smoking to be sure about that. So length of time, really hard to say, and probably quite inaccurate. But 5 to 10 years, that kind of range of time would be reasonable.
This podcast is funded by the British Society of Lifestyle Medicine. The BSLM is an independent charity set up to promote the role of lifestyle medicine in improving people's health and well-being. We empower healthcare professionals to support patients to prevent, manage, and even reverse many chronic conditions through education and connection to our like-minded community. Shiv and I are both members of the BSLM and certified lifestyle medicine practitioners. We authored our first course, The Fundamentals of Gut Microbiome Science, for BSLM. This podcast was created with the aim of improving education on the role of human microbiomes. For those keen on doing a deeper dive, please check out the course on the BSLM Learning Academy page. Listeners to this podcast can access a 10% discount with code MBM10. We're also able to offer all healthcare professional listeners 50% off the first year of membership, unlocking a free overview course and nearly £100 off annual conference tickets, using the code MICROBM in capitals. Now let's get back to the show. So we know now that pollution and smoking are definite impactors.
Yes.
What about vaping?
Vaping, watch this space. So we're just starting with collaborators a project around vaping. So Dr. Aaron Scott at the University of Birmingham is an immunologist, and he's looking at inflammation in the context of using either vaping or nicotine patches to help you stop smoking.
Okay.
So our contribution to that study will be looking at the microbiome in that context as well. I'd imagine it probably has an impact. I'd imagine it was probably a little bit different to cigarette smoke because it's a different range of compounds that you're being exposed to. I think the thing to remember with vaping as well is how variable it is, because it's not regulated sort of market in the same way as cigarettes and smoking, and there's a lot of flavor compounds that go in there, big variety of them. So what happens to them when they're heated, and how those interact with our own cells, all the bacterial cells, the microbiome that's there, is going to be unpredictable.
Yeah, and I think I've read one paper that has looked at the impact of vaping already, and found that those flavors and the flavors when they're initiated into lungs do create some inflammation, as you would imagine.
Yeah.
But the other thing is the heating element in the vape, because the heating element actually gives off a whole load of metals.
Yeah.
And you inhale a load of metals, and they've actually found quite a lot of different metals in the lungs, chromium and tin and aluminium and all sorts of things. And a lot of these metals are actually quite inflammatory as well.
Yeah.
Aluminium is very inflammatory in the lungs. So that's an interesting one as well, because and just the heat, I think, from vaping, because heat in its own right can create inflammation, but also DNA damage in cells, can't it?
Yeah, it's such a difficult topic, because one of the things I think concerns me as well is the age of people vaping is also much, much younger, and what we're actually talking about there are sort of developing lungs that are still developing, and causing damage to them early on is likely to store up issues for later. Smoke as a smoking cessation tool, it's a little bit more nuanced, because there you're talking about, is it better than cigarette smoke? and it's a relative thing. So if it's helping people to stop smoking, and while it might still be damaging, it's better than smoking, it's trying to measure a sort of relative risk there. But I think it's probably safe to say if you've managed to stop smoking by vaping, it would probably good to be trying to stop vaping after that as well, because nicotine in and of itself is not a great thing to be getting into your body.
Yeah, nicotine on an adolescent brain as well is very harmful. It can cause behavioral problems and mood problems.
Okay.
Interestingly, I looked it up. 10% of our adolescents are vaping now, and I wonder actually if that's even higher in my local kids' school as well. Because talking to my own children about how many kids in their year are vaping, and actually you're more likely to smoke if you start off vaping.
Yeah.
Yeah, as much as it's been used as a way of stopping smoking, it actually may initiate you towards smoking as well, which is not wonderful. I have my concerns about vaping, and I've seen young patients coming along with serious vaping habits, equivalent to 20 cigarettes a day. And whilst I agree they're probably safer than cigarettes, there's still a risk there, and I think that really needs to be ironed out. And I think our children need to be kept away from vapes as much as possible to preserve their developing lungs.
There's a difference there, isn't there, between somebody who's maybe in their 50s and has smoked their entire life and needs to get off cigarettes for their lung health. That's different to a sort of 12, 13, 14-year-old with growing, developing lungs who is starting vaping and hasn't tried cigarettes yet. They're two very different usage cases and two very different sort of outcomes probably as well.
That's smoking, vaping, pollution. What about volatile organic compounds? Have you come across these much? VOX?
I mean, so not in the context of the lung microbiome very much, to be honest. I think what we're often talking about with these things is lung inflammation. Mm-hmm. And that's going to be key for understanding the interaction with the microbiome, because I think one thing, we're always looking at chicken and egg with gut microbiome, with lung microbiome as well. What's causal? And we know that there's some pathogens that you can spread around, and they will infect you, and they will cause inflammation. They'll cause an infection. They'll cause inflammation. So there's examples of viruses like that. There's examples of bacteria like that. So one of the streptococci, Streptococcus pneumoniae, we can carry it, but also it can cause pneumonia. However, there are other situations where we might have some kind of inflammation caused by something else, whether that's external, like pollution, volatile organic compounds, or whatever. And then the bacteria already in your lungs are in a different environment to what they were in before. So we see some of the shifts in communities that we see in microbiome studies might just simply be a population of bacteria who are having to live now in a much more inflammatory environment. So they're dealing with a lot more immune system than they're used to dealing with on your average day. So their response might be to just disappear and die, or it might be to become more actively pro-inflammatory as well. So anything I think that causes inflammations in the lungs is going to have an impact on your respiratory microbiome.
Definitely. Whether
those changes are then part of the pathology of what then happens afterwards, it's hard to say. So volatile organic compounds, particularly bad for lungs. It's probably not a good thing to be inhaling all the time.
We should probably say what they are, actually.
Go for it. There's all sorts of different things. It's a huge category of chemical compounds.
So they're chemicals that vaporize at room temperature. So the sort of things that contain your VOX are your spray deodorants and your spray cleaning products, furniture, floor polishes, oven cleaners, detergents, dishwashers, dishwashing liquids. And these compounds, they, as you say, they're totally variable. And they've studied, there have been some studies on hairdressers that use more. So for example, hairdressers that deal with afro hair more, they use more of these compounds to straighten the hair. And they did have a difference in their lung microbiome, presumably through sputum samples, than the hairdressers that didn't use quite so many of those compounds. And I think Voxes or exposure to Vox have been linked to lung cancer.
Okay.
That I'm aware of. And there's few studies on that now. And women especially seem to be more vulnerable to the effect of Vox on the lungs. From a study, there was a study that was written in 2018, long-term effects of cleaning on the lungs. And it looked at a whole bunch of 6,000 adults between 1992 and 1994. And they looked at people who clean for a living and people who clean domestically. And women especially who clean for a living had a lot more disruption to their microbiome, and they looked at levels of inflammation. I think they're measuring FeNO. Is that right?
Oh, yes. I've heard of that one.
Yeah.
So this is the thing. So I'm not a sort of respiratory doctor. And I think that, so this sort of heads into the realm of occupational exposure and those kind of respiratory diseases. So they're often also less well studied than, either the chronic infective kind of lung diseases. So these sort of exposure diseases, because maybe they're harder to spot. So hairdressers, for example, it's a more dispersed group of people. They're not all working for one organization, and trying to, even noticing that they have particular sort of impacts on their respiratory health is hard work.
Mm-hmm. In this world that we're living in, we're obviously surrounded by these chemicals, aren't we? It's very hard to avoid them. They're in our lives in so many different ways. So it would be good to get more research into this.
I think so. Yeah, for sure.
And to get more understanding of, the harms from these things, because, we need to know if we are doing ourselves harms with our once a week clean, or if we should be, switching to, less harmful things, liquids, the preparations rather than spray preparations. What's the risk? If we're using deodorant, should we use liquid or, solid deodorants, or should we use spray?
Should we be rolling on or yeah.
And so I do worry about the impact of these chemicals on the lungs. And it makes a lot of sense from the reading I've done into aluminium and into various different chemicals that are in these products, that it would be creating inflammation there. So I think this would be, good to know a little bit more about. So let me know if you know anybody that's investigating Vox, because you're the guy who's going to find out.
I think the interesting thing there around microbiome, though, is like, what would the solution be? Is that a microbiome solution that we would be coming up with for that kind of thing? But actually, it's the solution there would be changing the way we clean, changing the cleaning products, not a sort of microbiome intervention. Maybe the microbiome is playing a part, but actually the solution there is probably not a microbiome-based solution. And that's interesting across the field, I think, as well, because, you see some people suggesting sometimes interventions that are microbiome-based, but for things that actually we can impact in different ways. And I'm in favor of choosing the right intervention there. So dietary stuff, for example, sometimes people reach for a probiotic or a live biotherapeutic, and you're like, well, maybe have we tested this against eating a banana once a day? Have we improving your fiber intake, or doing the sort of boring kind of 5-a-day interventions rather than necessarily going straight to a probiotic as your first line of things to do.
Okay, well, you're moving on to diet now. So that was one of my next things.
Yeah.
Okay, how does diet influence your lung microbiome?
I have lovely occasional arguments with. So we have a really famous microbiome researcher at Birmingham, Professor Lindsay Hall. So she works on the infant gut microbiome,
I know,
and obviously, and particularly sort of mother and baby pairs and preterm birth and things like that, and how that relates to particularly bifidobacteria. But we have a semi-regular conversation about dietary impact on the lung microbiome, and she's like, yes, of course there's going to be a massive impact, and I'm like, no, there definitely won't be, and probably the reality is somewhere between the two of those things. So I think the thing to remember is time scale and location of stuff. So sometimes diet can have systemic effects, but the important part of the interaction with the lung is that there are microorganisms right there next to those cells. And we're always thinking, so gut microbiome focus sort of, it influences the whole body. It's like, yes, but these organisms are right there next to these immune cells, being sampled by these immune cells, interacting directly with the epithelium. Gut microbiome is further away. It's in a different compartment. It's a different composition. So to have an impact there, those organisms have to be churning out stuff that goes through a lot of filtering into the bloodstream, around the body. Then it's a more distal effect. So what we're looking at there really is time, I think. So in shorter time scales, in the time scales of sort of catching an infection, being exposed to some pollution or something like that, local processes are going to be super important. The time scale of sort of a life or years or those kind of things over the period of time where you might affect a change in your diet, then probably that is going to have some sort of systemic impact as well. So she and I sort of have this conversation regularly, and I think really what we're talking about is time scale there. So if you change your diet, probably the impact is not going to be anything immediate. If you change your diet in such a way that you change your weight, you change your lifestyle, then maybe longer term we will see those things. Those studies haven't been done because they're hard to do in the gut, let alone...
They tend to be epidemiological. So they tend to be looking at, again, correlations, don't they? So people that eat more than 5 a day of fruits and vegetables...
well done then, firstly.
Yeah, tend to have lower... They tend to have lower rates of asthma, for example, or COPD.
Yeah.
And I've always thought that it did play a role. Personally, because I had 2 grandmothers who smoked for 50 years, neither of which got COPD. And they had a lot of plants in their diet. And okay, they were quite, they did enough exercise and things as well. But I often thought that their lifestyle played a role in that.
Yeah.
And that is very anecdotal. But I've actually used that process with patients and got them on higher fiber diets. And what I've noticed is my COPD patients have particularly low fiber diets when they're diagnosed, and I put them on a much higher fiber diet. And the last chap that I did, we don't have a lot of COPDs in our practice, but we had a guy who had quite severe COPD. This was about 8 years ago, and he came to me and he had a low fiber diet, not doing very much exercise. And I got him to adopt a high fiber diet and exercise really, really regularly. Mm-hmm. So it's a cumulative. It's a few different things that we're changing. But in that time, since in that 8 years, instead of nosediving like most of my COPD patients had done in the past, he's actually remained incredibly well and fit and had 1 infection in that time that I'm aware of. So he's remained very, very fit and healthy, and not got the number of infections that I was expecting that he would get, compared to other COPD patients. And the other little bit of evidence that I see as a doctor is the oral microbiome and the importance. So whilst you say that diet's going to have that sort of slightly longer effect, it's going to take longer for it to impact coming from the gut. the oral microbiome is going to change quite rapidly with diet change, isn't it? And the oral microbiome is going to impact
It's the
on
source
the
of
lung.
organisms. Yeah, 100%. It's so difficult, and it's really interesting because it's so hard in a human to make one change, because you're always, so if somebody's improving their diet, then maybe they're feeling a bit better, they get out a bit more, and then exercise becomes an impact, or even it turns out that they start running in a field and they're getting a lot more hay fever and it goes in the other way, or, all this kind of stuff. Oral microbiome for sure is linked to things like COPD. So, improved dental care. You've got less plaque, less organisms like that sort of heading down into your lungs. So there does seem to be a relationship there. The cardiac doctors will probably say don't brush your teeth too much, because there's another link between sort of endocarditis and sort of heart health and brushing your teeth too much, or doing too much flossing and removing this stuff which goes into your body and bloodstream and can link to heart disease in that way. So yeah, it's super complicated, particularly when we start talking about a human and their life. But
And I do wonder if those lactobacilli in nice milk fermented foods and things are going to also benefit the lungs just through the effect on the immune system. And
it is funny. So lactobacilli are sort of falling out of favor in the gut space as well. So people like the bifidobacteria are what people are sort of going after, but there's trends around this. At the moment, we don't even know whether the ones that you find in the lungs are the same species as the ones that we have in the guts. Mm-hmm. So again, at the moment, we're talking about a distal effect of those lactobacilli, even if there is one at all. Mm-hmm. And trials of probiotics are sort of fraught with many more failures than there are sort of success stories. Mm-hmm. It would be lovely to see some interventions in the lung space that acknowledge the existence of a lung microbiome to start with, and then maybe start to utilize it as a real tool for helping us with our health. I think that's where I'd like to see things go, but at the moment we're quite far off that.
Well, as microbiome medics, we don't push people towards probiotics very often, but we would push people towards nice healthy diet with fermented foods.
It's the banana. I think it's
fermented
banana.
foods regularly, and the Sonnenbergs in the US have really shown the impact of fermented foods on the gut and how beneficial that is. So we haven't touched on exercise in the lungs, and I think that's a really important area.
Okay.
So tell me about exercise in the lungs.
So it's not something, I think, again, I'm sort of thinking mostly about a sort of proxy here, which is respiratory physiotherapy, and shifting stuff up and out of the lungs. Mm-hmm. If you have a lung disease. Certainly that's been shown to have a really positive impact in cystic fibrosis. Mm-hmm. And it's part of normal therapy there for people with cystic fibrosis. If you can improve your sort of lung health and your in general, and exercise is one way to do that, then probably it's going to help. Prevention is a really strong word, but certainly improving your risk, I think. Again, we'll probably see that at the population level. Really hard to say what it's doing at an individual level mechanistically in terms of the lung microbiome. But I think numbers, fewer numbers, if you've got an increase compared to normal, is probably a good thing.
Yeah, as you say, there's probably so many different mechanisms by which exercise benefits microbes.
Yeah.
And, it depends whether you're exercising in nature or exercising, in a gym environment. Again, you're going to pick up different microbes and things, aren't you? And and you're going to have different impacts from fresh air and
if there's somebody in the corner who happens to have RSV or COVID at that particular time, then that's going to outweigh the impact of your your particular exercise regimen on that day.
Yeah, but hopefully, if you're exercising hard, then hopefully you're stimulating your immune system enough to be able to tackle those viruses that are probably in the air all the time anyway.
Yeah.
Okay, so exercise, good, and we know from epidemiological studies that exercise really helps with the reducing your likelihood of asthma, COPDs, all these chronic lung conditions, and of course helping reduce your chance of more serious lung conditions like lung cancer.
Yeah.
So they're all beneficial, but
I think we have to remember there, sorry to interrupt, that we do have to remember there the genetic components to these diseases as well. So, because sometimes I think, we sort of encourage groups of people to do things, but actually it's very hard for them because, they have a genetic predisposition to a particular thing. So asthma has a genetic component as well as an environmental. COPD, there's people with alpha-1 antitrypsin, so I'm a carrier of that, actually, and that gives you a much higher chance of getting COPD. Yeah. So exercise is a component for sure, but it's only going to take some people so far of the way.
Of course, and some genetic conditions are, your genes are your fate. With the case of cystic fibrosis, of course, you can't turn that around. But for a lot of genetic disorders, genes can flip and genes can change. And epigenetics is a very interesting thing where, your lifestyle actually can massively influence your genes, and you can turn on and off genes through, changing your lifestyle quite dramatically. And in lifestyle medicine, we learn a lot about epigenetics and how we can influence our genes through diet and lifestyle and, sleep and exercise and all of these things. So there's hope for the majority of us. But of course, if you have a serious genetic disorder, then unfortunately we can't often flip those genetics for you. So yes, it depends on the genetic disorder, doesn't it? Okay, all right, interesting. So if you have one message, Mike, before we go, about the microbiome that you want to get across to people, you want to spike their interest and obviously get them thinking about the lung microbiome, what will you say?
That's a challenge. What this always makes me think of is slightly bored students in my lectures, and I so how do we get them engaged? I think the thing with the lung microbiome is that it is real. It changes with disease, and we really need to understand it because there's real potential there to have some kind of intervention that makes conditions last less time. I would hope that actually we get some ways of predicting when we need to intervene through understanding the lung microbiome. That's where I'd really like to go, I think. So some sort of meaningful intervention and also understanding what interventions aren't worthwhile as well. Because again, interfering too much can be an issue too. So that's what I'm interested in particular, I think, in this space. And I'm hoping my sheer enthusiasm will carry other people along with me because I'm not sure whether the message does or not.
Well, the interest in microbiomes, human microbiomes in general, is just climbing constantly. Yeah. I'm just, I'm even seeing it in my primary and secondary care colleagues now, that more and more of them are taking note of microbiome science and want to learn about the gut microbiome and other human microbiomes. So, I think the interest is starting to peak now, and we just need to keep people's enthusiasm up and hopefully, more interest, instead of just thinking of the lungs in terms of physiology and, in terms of the diseases of the lungs, let's think a little bit more deeply and consider the microbes as well. And think about them and how there's this other element here that could be changing the dynamics of this important organ. And we all should be aware of these little organisms and how they're impacting our health. And we need to learn more, as you say, as to why that is. So thank you so much for coming on today. Good luck with all your research. What's next for you, Mike?
I think we're trying to understand what the bacteria in our lungs are eating, because they've got to eat something, and then we can tweak that, and then we've got a little tool to manipulate the communities. So that's one of the next things we're looking at.
Brilliant. Right. Well, keep me informed. Keep me updated.
Will do.
And take care. Nice to see you.
Bye-bye.
Thank you so much for listening to this episode of Microbiome Medics podcast. We really hope you enjoyed the content and we welcome your feedback. We'd love to hear any suggestions you might have for microbiome topics that you'd like us to cover. And we also appreciate listeners' questions and will endeavour to answer them in the next podcast.